Hello everyone, today we are going to talk about reproductive physiology. Let's talk about some important hormones. In our brain we have two glands, our hypothalamus gland and our pituitary gland. Our pituitary gland is divided into two lobes, the anterior lobe and the posterior lobe. We are going to focus on the anterior lobe. So our hypothalamus produces a hormone known as GnRH, which stands for gonadotropin releasing hormone. Gonado coming from the term gonads, which are the ovaries and the testes. So the gonadotropin releasing hormone is eventually going to stimulate the gonads to release hormones and to do specific tasks. But the gonadotropin releasing hormone does not stimulate the ovary and the testes directly. What it does is it stimulates the anterior pituitary gland, and once the anterior pituitary gland is stimulated, it's going to release two hormones. FSH, which is the follicle stimulating hormone, and LH, luteinizing hormone. Both hormones are produced in males and females. The FSH, when it's released in females, it's going to help the growth of ovarian follicles. And we're going to talk about those ovarian follicles when we are going to talk about the oogenesis process. FSH in males stimulates spermatogenesis. So spermatogenesis is the production of sperm in males. Luteinizing hormone in females, it's going to help with the ovulation process, and it's also going to help with the production of the sex hormones, such as estrogen, and it can also, in an indirect way, stimulate the production of progesterone, and we're going to talk about how. Luteinizing hormone in males helps with the production of testosterone. A quick review about mitosis and meiosis. Mitosis is the cell division that would result in daughter cells that are identical to each other, and they are identical to the parent cell. That's why we refer to mitosis as cell cloning. So if this is the parent cell, and this parent cell contains all the chromosomes, so it's a diploid cell, it's going to divide, and the result is going to be two daughter cells that are also diploid, and they are identical to each other, and they are identical to the parent cell. That's mitosis. And for mitosis to occur, we have to go through prophase, metaphase, anaphase, and telophase. Meiosis, on the other hand, we are still going to go through prophase, metaphase, anaphase, and telophase, but we are going to go through these stages twice. So we call it meiosis 1 and meiosis 2. In meiosis, the parent cell is going to divide once, and we're going to get two daughter cells that are now going to be haploid because they are going to carry half the number of chromosomes. So that's the first mitotic division. And then those daughter cells are going to go through a second mitotic division. And so the end result of meiosis, we are going to get 4 daughter cells. Those 4 daughter cells are going to be haploid. They're going to be carrying half the number of the chromosomes. and they are not going to be identical to each other, and they are not going to be identical to the parent cell. So in meiosis, we go through two divisions, meiosis 1 and meiosis 2. And so we have to go through prophase, and so we call it prophase 1, metaphase 1, etc. At the end of the first mitotic division, we get two haploid daughter cells. Then we enter into the second mitotic division. We refer to the stages as prophase 2, metaphase 2, etc. And at the end of the second mitotic division, we get four haploid daughter cells. So those are going to be carrying half the number of chromosomes. When we are going to make our gametes, which are our eggs and sperm, we go through meiosis. We actually go through both mitosis and meiosis, but meiosis is going to be pretty important because meiosis is going to result in haploid cells. So our eggs at the end of the day are going to only carry 23 chromosomes, and sperm are only also going to carry 23 chromosomes. So that if fertilization does take place, the chromosomes of the egg will combine with the chromosomes of the sperm, and we will make a zygote that carries the full set of the chromosomes, which are 46 chromosomes. In males, the sperm are produced within the testes, within those seminiferous tubules. If we zoom in on the seminiferous tubule and we look at it under the microscope, this is how the seminiferous tubule looks like. The sperm are going to be made, and they are actually going to be pushed into the lumen. The lumen is just the cavity. So they are going to be pushed into the lumen, the cavity, so that they can travel into this region right here, the testes, and then from there they're going to travel into the epididymis. And then if ejaculation is to take place, then the sperm is going to leave the epididymis and it's going to start traveling up the ductus deferens or the vas deferens. The process of making sperm is known as spermatogenesis, which starts at the age of 14 in males, and it's actually going to continue throughout the lifetime of the male. Every day, adult males make about 400 million sperm. Within the testes, in the seminiferous tubules, there are stem cells known as spermatogonia. Singular, we would call it spermatogonium. Those stem cells, they contain the full set of chromosomes. So they are diploid, or you can refer to them as 2N. Those spermatogonia cells are going to begin dividing when a male hits puberty. So when a male hits puberty, those spermatogonia or those stem cells are going to start going through mitotic division. As we said, mitosis results in two daughter cells that are identical to each other and identical to the parent cell. We refer to those daughter cells as type A and type B. Type A cells are going to remain within this region right here so that they can maintain the pool of the dividing germ cells. Type B are going to get pushed into the lumen like I showed you in the previous image. So they're going to be pushed into the lumen and they are going to become primary spermatocytes. Those primary spermatocytes are the ones that are destined to start dividing so that they can produce 4 sperm cells. Each primary spermatocyte, as you can see, it still carries the full set of chromosome. That primary spermatocyte is going to go through the first mitotic division, and you are going to get 2 haploid secondary spermatocytes. Those secondary spermatocytes are going to go through the second mitotic division, meiosis 2, and we are going to get 4 daughter cells. And those 4 daughter cells, as you can see, now they are haploid. So instead of 2N, we are just going to refer to the set of chromosomes as N to kind of tell you that now they are only carrying half the number of chromosomes. Those spermatids right here are non-motile, so they can't move. So they will have to go through a process known as spermatogenesis so that they can start looking like a mature sperm with the head and the tail. So that's the process of spermatogenesis right there that a male will go through every day. So the process of spermatogenesis, so we're not going to go over it in too much detail, but as you can see, it takes about 24 hours really for the sperm to start becoming mature and look like the sperm with the head and with the tail. The head of the sperm contains the DNA. So as I said, it's only going to carry half the DNA. And there is this region right here known as the acrosome. And the acrosome, it's actually containing hydrolytic enzymes that's going to enable the sperm to penetrate the egg during fertilization. That mid piece of the sperm is going to contain mitochondria to give the sperm energy so that to allow the sperm to basically move to reach the egg. The tail is basically a flagellum, so it can help the sperm swim and move towards the egg. Within the penis of a male, there are 2 erectile tissues. There is the corpora cavernosa and there's the corpus spongiosum. During sexual excitement, erection is going to be triggered by the parasympathetic reflex. And that's going to promote the release of nitric oxide. Nitric oxide is going to relax the smooth muscles in the penile blood vessels within the erectile tissue. And so when those arterioles are going to dilate, the erectile tissues are going to be engorged with blood. The expansion of those erectile tissue is what's going to cause the erection. The expansion of the corpora cavernosa is going to compress actually on the veins that are located there. And when it compresses on those veins, it's going to prevent the blood from flowing backwards, maintaining the engorgement and therefore maintaining the erection. When the corpus spongiosum, which is right here, deep, closer to the urethra, when the corpus spongiosum expands, again due to the corpus spongiosum being engorged with blood as well, its main job is actually to keep the urethra open during ejaculation. Because the semen is going to pass through the urethra, so we want to maintain the urethra open. The process of ejecting the semen out of the body, we refer to it as ejaculation. The semen is basically a combination of both sperm and secretions produced by the accessory glands such as the prostate gland, the seminal vesicle gland, and the bulbourethral gland. So ejaculation is actually under sympathetic control. So we said erection is under the control of the parasympathetic nervous system. Ejaculation, on the other hand, is under the control of the sympathetic nervous system. During ejaculation, the ductus deferens is going to start to contract, propelling the sperm down the ductus deferens, and then as the sperm is making its way into the ductus deferens, it's going to get into contact with those accessory glands, and those accessory glands are going to push out the secretion, so that eventually the sperm, along with all of these secretions, will exit the body via the urethra. Now, during ejaculation, the bladder, so here is the urinary bladder, the urinary bladder sphincter is actually going to constrict and close to prevent any urine from expelling out of the body along with the semen. Another reason why the bladder sphincter is going to constrict, so that it will prevent any reflux of the semen from actually flowing backwards into the bladder. And the ejaculate that's going to be ejected out of the body can actually be ejaculated at a speed of 11 miles per hour. In females, the production of eggs is going to take place within the ovaries. When a female hits puberty, each month she's going to be releasing an egg out of the ovary into the uterine tube or the fallopian tube. If a sperm meets an egg while it's in the fallopian tube, fertilization might occur. If fertilization does occur, then the fertilized egg is going to move its way down to implant itself into the uterus, and this is where the embryo is going to grow. If fertilization does not take place, and so therefore no pregnancy, then the egg that's going to be released along with the inner wall of the uterus is going to shed. And that's when we refer to it as the menstrual cycle that would take place each month. And so the uterus is going to shed part of its endometrium. So the uterus has 3 layers: the perimetrium, the myometrium, and that inner wall, the endometrium. It's only going to shed part of its endometrium, and it's going to exit the body through the vagina. This is an image of the ovary. So within the ovary, there are tiny sac-like structures known as ovarian follicles. The follicle is going to contain an immature egg known as an oocyte uh, inside of it. So those oocytes are going to be encased by cells. We refer to them as follicle cells. If there's one layer of those cells, we call them follicle cells. If there are multiple layers of cells surrounding an oocyte, we refer to them as granulosa cells. And those follicles are going to grow along with the oocyte inside of them. So first we get a primordial follicle, and it usually has just a single layer of follicle cells surrounding the oocyte. And then that primordial follicle is going to start to grow, and those follicle cells surrounding the oocytes are going to grow. And so we start calling them primary follicles, and then that primary follicle is going to grow and it's going to give you the secondary follicle, etc. And so then you get this like mature follicle right here known as the vesicular or antral follicle. And so you can see the cells around the oocytes grow. So instead of having a single layer, I mean, by the time you reach the vesicular layer, you actually have multiple layers surrounding the oocyte. And then the oocyte in there also grows at the same time. Um, and this vesicular follicle right here usually contains a cavity that's filled with fluid. We refer to it as antrum. And then each month, when a female, after a female hits puberty, each month the female is going to be releasing um, and ejecting an oocyte from its ovary. And we refer to that as the ovulation process. So when the oocyte is going to rupture out of the ovary, the ruptured follicle cells that were surrounding those oocytes are actually going to be transformed into the corpus luteum. The process of making eggs in a female is known as oogenesis. This slide right here is showing you the mitotic events. So it's showing how the stem cells within the ovary are going to start dividing, and how many chromosomes are they going to be carrying as they divide. This slide right here is showing you the follicle development, and those are basically the cells surrounding the egg. And we're going to call the egg an oocyte. So there are cells that are going to surround those oocytes, and those cells are going to keep growing. And so basically those two events right here are taking place at the same time. During the fetal period of a female, so before birth, there are stem cells in the ovary known as oogonia, or singular, you would refer to it as oogonium. Those stem cells are going to multiply by mitosis so that they can just clone themselves and make more of themselves. And so as you can see, they are still diploid. They are still carrying the full set of the chromosomes. Now, when the stem cells, they multiply and make more of themselves via the process known as mitosis, we start calling them primary oocytes. Those primary oocytes, they are going to be surrounded by a bunch of cells known as follicle cells, and so we call the whole thing the primordial follicle. So the primordial follicle is basically the primary oocyte surrounded by a single layer of follicle cells. That primary oocyte is going to begin its first mitotic division. So meiosis 1, um, when um, when the female is now an infant or a child, that primary oocyte is going to start meiosis 1, but it's going to actually freeze during prophase 1. Um, so it's not even going to complete prophase 1, it's just gonna freeze right there. By birth, we say that females are going to have their lifetime supply of those primary oocytes. By puberty, there are actually going to be around 300,000 oocytes that will be remaining. This primary oocyte right here, during infancy and childhood, they are still surrounded by the follicle cells, and we still refer to the whole thing as the primordial follicle. Now, before puberty, any primordial follicle that was within the ovary would actually die. So that's why we say by the time a female hits puberty, she will have remaining around 300,000. Because before puberty, we actually started with way more, but as I said, they were dying. So at puberty, a female is going to start producing the gonadotropin hormone, and the female is going to start producing FSH and LH. And the FSH is going to actually start rescuing some primordial follicles from dying each month. So one of those rescued follicles, when a female hits puberty, is actually going to be selected to become the dominant follicle and to actually start completing meiosis 1. So as we said, during childhood, the primary oocyte is going to begin meiosis 1, but as we said, it freezes. And it's going to stay frozen at prophase 1 until a female hits puberty. Once a female hits puberty, that primary oocyte is going to go ahead and finish off meiosis 1. And when it finishes off meiosis 1, it's actually going to produce 2 haploid cells. This one right here is that smaller one. It's called the first polar body. And then this one is now going to be called the secondary oocyte. So as the primary oocyte is finishing off its meiosis 1 and it's giving you those 2 haploid cells, at the same time, the cells that are surrounding that primary oocyte is going to start to grow. And so now, instead of calling it primordial follicle, we are going to call it primary follicle. And then that primary follicle, as you can see, the cells around the oocyte is starting to grow even more, and so we refer to it as the secondary follicle. And then it starts to grow even more, and we start actually having a full, a fluid-filled cavity known as the antrum. So we refer to it as the vesicular or antral follicle. So within that vesicular follicle, this is where we're going to have our secondary oocyte. As I said, those 2 events are happening exactly at the same time. So in here, you will have your secondary oocyte. By the way, those polar bodies are not going to contain cytoplasm or organelles, so those polar bodies are not really going to play any major role in the process of, like, fertilization further on. So these are going to be the important ones, the secondary oocytes. So the secondary oocyte is actually going to go through the second mitotic division. As we said, with meiosis, we go through meiosis 1 and we go through meiosis 2. So the secondary oocyte is going to go through meiosis 2, and it's going to freeze at metaphase 2. So the secondary oocyte is going to freeze at metaphase 2 stage. Now, during ovulation, even though the secondary oocyte is frozen, during ovulation, what's going to happen is the LH hormone is going to increase tremendously. And so when you get that LH surge, that's what's going to trigger ovulation. And so what's going to happen is that this vesicular follicle, which is now carrying the secondary oocyte that's frozen in metaphase 2, it's going to rupture open, as you can see right here. It's rupturing open, and it's going to release that secondary oocyte. And that secondary oocyte is going to be exiting out of the ovary. So it's going to completely exit the ovary, and it's going to start entering into the fallopian tube. And that secondary oocyte, if it gets ovulated, if a sperm meets the secondary oocyte in the fallopian tube, then that secondary oocyte is going to go ahead and complete meiosis number 2. But if a sperm never penetrates that secondary oocyte, then the secondary oocyte is going to deteriorate, and eventually it's going to be lost when the female has has the menstrual cycle. So that secondary oocyte is only going to finish off meiosis number 2 if a sperm penetrates that secondary oocyte. And if that happens, then after finishing off meiosis number 2, the secondary oocyte is going to yield a second polar body and an ovum. And the ovum is going to go ahead and carry now half the number of the chromosomes. What's going to happen to all of these follicle cells? Remember how we said the follicle cells surrounding the oocyte, it's going to rupture, and we're going to have the secondary oocyte rupturing out of the ovary. Well, surrounding the oocyte, you still have the follicle cells, or when you have a bunch of them, by the way, we refer to them as granulosa cells. So a bunch of follicle cells, we refer to them as granulosa cells. What's going to happen to all of these granulosa cells surrounding the oocyte? They turn into something known as corpus luteum. So the ruptured follicle or granulosa cells become the corpus luteum. In a female's lifetime, as we said, a female is going to have 300,000 primary oocytes. But in a female's lifetime, fewer than 500 oocytes will actually be released out of the ovary. And so this summarizes the process of oogenesis. And as I said, at the same time, the oocytes are dividing and going through meiosis 1, meiosis 2, etc. The cells surrounding the oocytes are growing at the same time. This graph right here shows the levels of the FSH and LH hormones within our plasma. This graph right here shows the ovarian cycle, so the events happening within the ovary. And then this right here is showing the levels of our estrogen and progesterone in our plasma. And then the last graph right here is showing the phases of our uterine cycle. So basically the stages or the steps that are taking place within our uterus. So let's go ahead and start talking about our ovarian cycle. So the steps happening within the ovary. The ovarian cycle is divided into two phases, the follicular phase and the luteal phase. And in the middle, we're going to have ovulation. So the follicular phase is going to go from day 1 to day 14, and the luteal phase is going to go from day 14 to 28. And ovulation happens on the 14th day, right in the middle. During the follicular phase, the reason why it's called follicular, that's because this is when we are going to have our follicle cells growing. So during the follicular phase, those primordial follicles, as we explained previously, they're going to start growing into the primary follicle, then into the secondary follicle, then into the vesicular follicle. And the more follicle cells grow around the oocytes, and you have more layers of them, we start calling them granulosa cells. So by the time we reach the vesicular follicle with the filled fluid cavity right here, the antrum, we've kind of like reached right here, and you can see that dominant follicle, the vesicular follicle right here. You notice that all of a sudden the LH hormone starts to increase tremendously. And so when you get that LH surge, that's what's going to cause the rupture of the vesicular follicle. And so when it ruptures, what's going to happen is the secondary oocyte is going to rupture out along with the granulosa cells that are surrounding it. And so right here, ovulation is going to take place around day 14 when the LH hormone starts to increase. By the way, during the follicular phase, those dominant follicles are going to start releasing estrogen. So you can see right here that during the follicular phase, the estrogen level is starting to increase and increase furthermore. That's because those dominant follicles, those granulosa cells, are actually releasing estrogen in the plasma. After ovulation comes the luteal phase. And so during the luteal phase, this is where the granulosa cells surrounding the oocyte, those granulosa cells that have been ruptured along with the secondary oocyte, they start forming the corpus luteum. So the corpus luteum, we don't have an oocyte inside of it. This is just some of the granulosa cells that were ruptured along with the oocyte. They are going to become the corpus luteum. Corpus luteum actually means yellow body, and the corpus luteum is actually going to start releasing progesterone. That's why you see right here during the luteal phase, our progesterone levels, the progesterone levels in females, it's going to increase because of the corpus luteum. And it also releases some estrogen as well. So you can kind of see the estrogen level also increasing. Now, if no pregnancy takes place, then the corpus luteum is going to degenerate in about 10 days. And we get a scar known as the corpus albicans. Albicans just means the white body. If pregnancy does take place, then the corpus luteum is going to remain until actually the placenta grows and until the placenta is ready to make its own hormones. And so the corpus luteum lasts for about 3 months if pregnancy does take place. Now, the menstrual cycle, or we refer to it as the uterine cycle, this is what's going to happen in the uterus. So the uterine cycle, we divide it into the menstrual phase, the proliferative stage, and the secretory phase. The menstrual phase takes place like day 1 to 5, and that's when a female is having her menstrual cycle, and she's shedding off part of the uterus, which is part of the endometrium of the uterus. And that's during the menstrual stage. This is when the estrogen and progesterone levels are pretty low, and the FSH and LH are also pretty low. Now, day 6 to 14th, that's going to be the proliferation phase. And the reason why it's called, again, proliferation, that's because during that time, the follicles are growing. And so you can see the estrogen levels are growing because that's when the follicles are growing right there in the ovary. But in the uterus, what's happening during that stage is that the endometrium layer of the uterus is actually rebuilding itself. And it's rebuilding itself under the influence of that rising estrogen level. And during that stage, the cervical mucus is actually going to start becoming less thick to facilitate the sperm passage into the uterus. Because as you can see, the secondary oocyte would have been ovulated. So that's why during that stage you want the cervical mucus to become less thick to make it easier for the sperm to reach the egg. So at the end of the proliferation phase around day 14, that's when ovulation is going to occur. Again, because around day 14 we are going to have the LH surge. Then comes the secretory phase, or we can refer to it as the post-ovulation phase. And during that phase, you can see that the endometrium of the uterus is preparing itself for an embryo to be implanted. So during that stage within our plasma, you can see that our progesterone levels are increasing. That's because the corpus luteum is releasing all of that progesterone, and with that progesterone surge, the endometrium grows. So that progesterone is actually going to cause the blood vessels in the endometrium to grow furthermore. That progesterone is going to cause the glands within the endometrium to start enlarging and releasing nutrients within the uterine cavity so that it can sustain the embryo. And then the cervical mucus actually starts to become viscous one more time, thick one more time, to prevent any further entry of any other sperm or any other pathogens. So you can kind of think of it as that the progesterone during this stage is playing a role in keeping the uterus private in an event that an embryo has already started to implant itself. And the rising progesterone levels within your plasma is actually going to prevent the LH levels from rising. And that would be important because you don't want to have another ovulation if the embryo has already started to implant itself within the uterus. Now, if fertilization does not occur, then the corpus luteum, as we said, is going to start to degenerate, and therefore the progesterone levels are going to drop, and so therefore the endometrium is going to start to deteriorate, and it's going to start shedding, which means we are going to go back to the menstrual phase and the cycle repeats itself.